Today we will discuss about southern blotting. Means what is southern blotting and how do we perform the southern blotting technique. Transfer of macromolecules from gel to membrane is known as blotting, and that macromolecules can be DNA, RNA or protein, and that membrane can be nitrocellulose membrane or nylon membrane, which have the ability to trap or absorb that macromolecules. If it is DNA, then it is called southern blotting, and if it is RNA, then it is northern blotting, and if it is protein, then it is known as western blotting. But today we will discuss about southern blotting. Just follow from step 1 to step 7. Suppose this is an eukaryotic cell, and first we have to isolate the genomic DNA from that cell, and the genomic DNA contain the target DNA or the gene of interest, which is known sequence, and the nearer to this target DNA is also known sequence. So we can choose or make the restriction endonuclease accordingly. Then go for restriction enzyme digestion. By treating the genomic DNA with restriction endonuclease, it can cut the genomic DNA into different fragments of different size. Then go for agarose gel electrophoresis. By putting this fragmented DNA into an agarose gel electrophoresis, so that this fragmented DNA can be separated on the basis of size. If the size of the DNA is large, its movement is very slow, and if the size of the DNA is small, then its movement is fast, and as the DNA is negatively charged, so it is always moved towards positive electrode. Then go for visualization, but from the binding pattern we cannot identify our target gene. For identification of target gene, we have to go for hybridization technique, but hybridization method is not possible on agarose gel, because agarose gel is very fragile in nature, and in a hybridization process we have to go for various fixation process, washing process. But if you do all these things on agarose gel, then it will break. So that we have to make a replica of agarose gel, and we will make a replica of agarose gel by using nylon membrane or nitrocellulose membrane, which can exactly make the replica of agarose gel. Before go to hybridization, first you have to convert this double stranded DNA into single stranded DNA, because hybridization process is not possible on double stranded DNA. Then just put this gel on a alkali solution for 2 hours. That alkali solution is sodium hydroxide. Then this sodium hydroxide can break the hydrogen bond between the nucleotides and convert this double stranded DNA into single stranded one. Then just take a plastic tray and put some transfer buffer into it. Then put a sponge. Then above the sponge put some paper sheet. Then above paper sheet put a gel, and above the gel put a nylon membrane. The size of the nylon membrane and gel must be equal, because the interaction between nylon membrane and gel is very important. Then put some paper sheet above to the nylon membrane. Then put some paper towel above the paper sheet. Then finally put a weight. Due to that weight, it creates a pressure, so that the interaction between gel and the nylon membrane takes place, and due to the capillary action, the buffer starts to move from the bottom towards the top, and during this movement of the buffer, the DNA from the gel move towards the nylon membrane also, and the DNA is negatively charged and the nylon membrane is positively charged. So when the buffer moves, during that time the DNA also moves, and that DNA get trapped inside the nylon membrane. After that we have to remove that weight, paper sheet, paper towel, and take the nylon membrane for further fixation process. And fixation process can be done by either heating or by giving UV light. After the fixation process, we have to put that nylon membrane in a probe solution for probe hybridization. Probe is a short stretches of the nucleotide having a detector. That detector may be radio labeled or fluorescent. After that, sorry, when we put that nylon membrane into the solution, then the probe go and bind with its target gene, because probe have the complementary sequence to this target gene. And then we have to take that nylon membrane into an autoradiograph, and if that probe bind to its target gene, then it generates light. Then the position of band can be identified from the cell, and cut the band from the cell and store it for further analysis.